Hello, everybody. We're just waiting a few minutes for everybody to register and join the room, and then we are going to get started very soon. All right, I think we can start. So welcome everyone, and thank you for joining today's webinar with Visiopharm and Molecular Instruments. My name is Leticia, and I will be your host for today's session. So we have an exciting session ahead, actually featuring 3 experts who will share insights on enhancing RNA-ISH spot detection with dual channel staining and AI analysis. So I want to welcome our speakers, which are Randy Chen, Director of R&D at Molecular Instruments. Then we have Dan Winkowski, Senior Technical Sales Specialist at Visiopharm. And finally, Regan Bird, SVP Research Commercial Strategy Deployment at Visiopharm. And just before we start, quick reminders. There will be a live Q&A at the end of the session. So if you have questions, you can submit on the chat. And this session is being recorded. So you have a chance to rewatch. We'll share with everybody at the end. So I guess we can just get started now. Enjoy.
Hi, this is Randy Chen, and I'm the Director of R&D at Molecular Instruments. Hi, this is Randy Chen, and I'm the Director of R&D at Molecular Instruments. This will be a joint presentation between MI and Visiopharm on the topic of enhancing RNA-ISH spot detection with dual channel staining and AI analysis. I will be representing MI talking about HCR RNA-ISH assay, and Dan and Reagan will be representing Visiopharm discussing how RNA-ISH data can be analyzed using Visiopharm software. In this presentation, I would like to explain how HCR-enabled RNA-ISH works and some of the challenges that users face in qualification of RNA-ISH probes. And finally, I'm going to introduce the concept of DCV, which is an acronym for dual channel validation. And I'm going to show how these challenges can be addressed by DCV. HCR stands for hybridization chain reaction. It is an amplification mechanism that's at the core of every molecular instrument's products. And the secret sauce of the amplification is that these HCR amplifiers are sequestered in hairpin conformation, and each of them is conjugated to a fluorophore or an hapten. And they do not interact with each other in solution unless initiators are present. And initiators are short DNA sequences that are right next to the probe binding sequence. And after probe hybridization, the initiator I1 opens H1 amplifier. The initiator sequence in the opened H1 amplifier will open H2 amplifier. H2 amplifier contains the same sequence as I1, which causes the next H1 amplifier to open And the process repeats itself that leads to the formation of fluorophore or hapten label HCR polymer. One of the major features of HCR assay is that the largest component of the assay is the HCR hairpins, which are only about 1.5 kilodalton. And this is significantly smaller than RNA-ISH amplifiers of the other RNA-ISH method. And this feature eliminates the need for harsh protease digestion, which can negatively impact co-detection of protein targets. And because HCR assay is protease-free, it allows negative compatibility with IHC targets. An image here shows the co-detection of polar 2A as RNA and membrane marker as protein in FFPE human lung cancer. And sometimes, as a membrane marker, DAB can overpower the ISH signals during data analysis. So user can choose a more translucent chromogen, such as green or yellow. This example shows a very nice cell segmentation using yellow chromogen as the membrane marker. Again, the protease-free assay allows membrane and nuclei features to remain intact. Dan from Visiopharm later on will show us, using their software, how we can better do cell segmentation and allow more accurate dot counting using HCR RNA-ISH assay combined with membrane marker. And before diving into dual channel validation, it's important to understand some of the challenges that we face to qualify RNA-ISH probes using conventional methods. So one conventional method is bulk RNA sequencing for whole tissue transcriptomics. This method can tell you the transcript abundance, but the downside is you don't know where the targets are expressed. In other words, you lose spatial information for the target of interest. And RNA detection in control tissues is another way to show if your probe is binding to a target of interest. However, it's not always easy or even possible to find positive and negative control tissues for every RNA target that you're trying to detect. Another common way to qualify RNA-ISH probe is qualification with IHC marker. In this example of ERBB2 detection, we see a positive correlation between protein and RNA abundance. However, if you look at the images on the left, how would you interpret the result in this example where you have RNA-ISH staining, but with an IHC score of 0? And moreover, from literature, we know that protein and RNA expressions don't always correlate. And it's not always possible to find the RNA counterpart of primary antibodies commercially. And so what do we do when validated antibodies are not available? And what do we do when we question the validity of the ISH signals? And we believe the dual channel validation is the answer to these questions. The way DCV works is, first you start with 2 independent sets of probes, but they both target the same RNA of interest. Then you run through the DCV assay, resulting in signals into separate channels. And the degree of co-localization and correlation of signal intensities will allow us to validate probe specificity as well as make inferences about the transcript abundance. This validation method is standardized, meaning you follow the same procedure for every probe you want to validate. And it's self-contained because you are using the probe itself to conduct the validation. Just to illustrate the methodology of DCV with an actual example, here we have a GeoPure probe set. First, you split the probe set into two groups and detect the same target redundantly in different fluorescent channels. Then you run these raw images through a script, and that will give you the colocalization factor and correlation coefficient. And based on these numbers, it results in a DCV score of 3+. And the reason DCV can be used to verify probe specificity is that if signals from two different channels are detected at the same location, we are very confident that the co-localized signals are real and specific, because it's extremely unlikely for two false positive signals to co-localize at the same location. And a good analogy would be if you consult two different doctors and both give you the same diagnosis, then you will be very confident that the diagnosis is correct, because it's unlikely that they both give you the same false diagnosis. DCV scoring guide is developed internally along with experts who are routinely doing RNA-ISH, and also with pathologists who are making diagnostic decisions based on the expressed signals. The score ranges from 0 to 3+. Score of 0 doesn't mean the probe is not working. It just means it hasn't gone through the DCV process. And having a score of 1 tells us the probe set is sensitive and specific. And having a score of 2, 3, and 3+ enables us to make inferences about target abundance based on the observed signal intensity. And it's important to know that having higher score doesn't imply higher sensitivity or specificity. Having higher score just means you can make more precise inference about target abundance based on observed signal intensity. So this is a pair of raw fluorescent images of mouse polar 2A in two separate channels. After running the raw images through a DCV script, it resulted with a score of 3. And polar 2A is commonly seen as a low abundance housekeeping gene. And then later on, we'll demonstrate dot counting with this particular target. We have also validated a number of clinically relevant targets. This one shows the validation of high-risk HPV probes. And we know HPV is associated with development of various forms of cancer. So it's critical that we validate the probe specificity before giving to users who are making diagnostic decisions. And we had a user who ordered a probe that targeted LRRC15, which is found to be highly expressed in tumor stroma. And we were asked to validate this probe on the tissue samples that he provided. And we were able to validate the probe set on all of the tissue sample types that he provided. And I'm only showing one here with human breast cancer tissue. And that resulted in a score of 3. And this illustrates that DCV can be applied to any probe set and on any FFPE tissue sample types. And thank you all for your time. Now I'll hand it over to Dan to dive into the analysis of HCR Pro images using Visiopharm software.
Thanks for the presentation, Randy, and thank you for the introduction. Hi, everyone. It's a pleasure to be here today to talk about a major challenge in the fast-paced world of spatial biology, accurate cell segmentation as it relates to spatial transcriptomics, and specifically how AI-driven methods, particularly membrane-aware approaches, are changing the game. As many of you know, multi-omic tissue imaging is rapidly advancing, allowing us to analyze gene expression in spatial contexts. One of the persistent challenges in multi-omic tissue analysis is dot detection, the process of assigning RNA-ISH signals to individual cells. If we can't define precise cell boundaries, we risk drawing inaccurate biological conclusions. So today, I want to walk you through why this is a problem, the limitations of current methods, and how AI-driven segmentation using membrane stains offers a powerful solution. Okay, so let's dive in. To understand why segmentation is so important, let's take a step back. When we analyze gene expression in tissue samples, we're dealing with RNA signals scattered throughout the tissue. But to make sense of this data, we need to assign each RNA dot to its correct cell. So what's the problem? Cell boundaries aren't always clear, especially if we rely on nuclear staining. As a result, traditional segmentation methods can fail due to misassigned signals or reduced sensitivity. This lack of precision presents a major obstacle in transcriptomics, making it difficult to extract meaningful, reproducible insights. So, for example, in the image on the top right, you can imagine the challenge of linking dots to specific cells, especially when you cannot confidently define the cell boundary. So, what's the solution? As we just heard from Randy from Molecular Instruments, they've developed a dual stain method that identifies not only the cell nucleus and RNA targets, but also stains for the membranes so that the cells can actually be identified. This helps guide how the cells should be divided and where the cell boundaries are located. This is where Visiopharm's discovery platform can come in. We offer an AI-driven platform in which we can train an AI to segment cells based on nuclear and membrane stains and correctly identify cell boundaries in your spatial transcriptomic workflow. This ultimately can lead to reliable, reproducible, interpretable results. Now, we worked with Molecular Instruments team on a project in which we applied the currently available analysis methods for RNA dot detection to their images and compared the results. And here's an overview of what those methods that we used were. First, manual annotation. Here, our human observers manually identify cell boundaries based on visual interpretation. While this can sometimes be accurate, it's extremely time-consuming, highly subjective, and suffers from a high amount of variability. The same image analyzed by two different experts may result in different segmentations. Even the same image analyzed twice by the same observer isn't going to be exactly the same. This lack of consistency is a major limitation, especially in large-scale studies. The second method that we tried, or we applied, was our pre-trained AI nuclear detection algorithm. Now, this is a much more automated approach where our pre-trained AI is used to identify nuclear staining and cell boundaries are estimated. In this case, the AI model finds the nucleus, expands outward a specified distance, and produces a rough approximation of the cell shape. This method is faster and more consistent than manual annotation, but also is not without its drawbacks. And I'll touch on those later. Finally, we developed a membrane-aware AI in our discovery platform. And this method takes advantage of both the nuclear stain and the membrane stain in the tissue, allowing the AI to learn the true cell boundaries for the cells in the sample. By incorporating both nuclear and membrane signals, we get a much cleaner definition of cell edges. So that's a brief overview of the project, and let's take a look at these approaches each of which in a little more detail. So first up is the manual approach. We asked a set of colleagues to manually identify cells in the same field of view, and the way they did this was just to place a number over the cells in the image. They were each provided the same field of view and then identified the cells by their reporting by placing a number over the cell. So exactly what the numbers mean or where they are isn't necessarily critical, but what is critical is a simple number of the count that each observer found, the count of cells that each observer identified. And as you can see, if we tally those numbers up for each observer, there's a wide range, right? There's anywhere from about 30 cells to 50 cells, and that's a big difference. With such a large range in the data, there's also some considerations for the downstream results. Remember, we're ultimately trying to count the dots in each cell, and we have a big difference already with just the cell counts. But some of the other considerations are how long did it take to get to the results, how accurate are the results, and what was the ground truth that each person used? Presumably that's slightly different for each observer. Are the results variable? Well, the cell counts indicate that, but also, what if we ask the observers to do it again on the same image? Would we get the same answer? Finally, all these factors speak to whether the results are reproducible. So there are some obvious drawbacks to this method and probably many more, but let's just say that this is probably not the best course of to pursue for analyzing these types of data. Okay, next up is our pre-trained AI model for nuclear detection. So this is by far a much more automated approach where our AI is trained to detect hematoxylin staining and estimate where the cell boundaries are located. And here's what that output looks like. So the estimation of the cell boundary is simply achieved by expanding from the nucleus outward and producing a rough approximation of the cell shape. As I mentioned, this method is faster and more consistent than the manual annotation, but it too has some significant drawbacks. It makes a lot of assumptions. It assumes uniform cell sizes, uniform cell shapes. Moreover, cells without a visible nucleus might be ignored. Upon evaluation, if we look at the outlines of these cells, we're doing a really good job finding the nuclei. But if we examine the outlines created with this method, quite often they fail to match the actual boundaries of the cells. Sometimes the boundaries drawn here cross over an actual membrane as shown by the staining, so that's obviously a concern. Moreover, we're missing many objects that strongly resemble cells but don't have an apparent nucleus in the plane of section. Ultimately, this can lead to uncertainty about which dot belongs to which cell and can greatly influence the results and interpretations of the study. Last up is our membrane-aware AI that we developed in our discovery platform. Now, this method takes advantage of the nuclear and membrane stains and allows the AI to learn the true cell boundaries based on ground truth coming from the chemical stain. So let's take a look at some of the results using this method. So here are the cells identified using this method. You'll notice right away that each labeled object closely follows the full morphology of the underlying cell objects in the image, which will therefore build our confidence in our ability to assign dots to specific cells rather than wondering which dot goes with which cell. Notably here, I'm showing only the cells that are identified to have a nucleus. There are many objects on the image that are not labeled that seem to have been missed, but they still strongly resemble the shape of a cell, especially because they're bounded by membrane stain. So as I mentioned earlier, with this method, we actually can identify those objects and label them as putative cells. And the user is able to decide whether they want to keep them as cells or not. It is up to the individual as to whether they want to include the cells lacking a nucleus or whether they want to discard them or maybe just analyze them separately. But the important point here is they can be captured by this analysis approach and are available at this point in time in the workflow and can be part of the measurements. And so by incorporating both nuclear and membrane signals, we get a much clearer definition of what the cell edges are, as you can see here with the way the labels are just created to be outlines. And obviously this approach offers several key benefits. We get more accurate dot assignment, so RNA signals are correctly attributed to individual cells. There's greater reproducibility with any automated solution. You're going to get the reproducibility that we're looking for, and remove the variability that might exist with other methods like the human annotation method. What I will also add here is that with this staining pattern and these staining conditions, this algorithm can work across different tissue types, making it highly adaptable. And to that last point, you might be wondering whether the solution is custom to the specific project or whether it can generalize across tissues. And the answer to that is yes, it can generalize, primarily because the algorithm depends only on the hematoxylin and IHC membrane stain. Here, I'm showing the performance on a few different tissue types that we tested, and obviously we'll continue to optimize in the future in collaboration with the molecular instruments team to create a robust solution for this. Okay, so here I'd like to start wrapping up my talk with a visual comparison and the methods we discussed today and if we just go from the image on the left to the image on the right, we're certainly advancing from a manual approach to an automated approach for dot detection. In addition, we're moving from an imprecise solution to a precise outlines of cell objects in the tissue. And finally, from left to right, we're moving from variable to highly reproducible, right? So all those things are benefits as we move from left to right in this sequence of methodologies. And now that we've compared these approaches, let's talk about the implications and why this advancement is so important for spatial biology research. So first, this method will lead to better biological interpretability. So when dots are assigned accurately, downstream analyses such as identifying cells, cell types, and gene expression mapping are much more reliable and make a lot more sense. It's scalable, so large-scale studies require automation and reproducible segmentation methods. Our membrane-aware AI allows researchers to apply this in batch mode where they can process large amounts of data rather efficiently. And finally, the robustness across tissues is an advantage because unlike nuclear expansion methods, which may work in some samples but fail in others, this membrane-aware AI is versatile and can work across diverse tissue types and conditions, at least at this time. So we're looking forward to expanding that capability too. Okay, so in summary, precise cell segmentation is critical for spatial transcriptomics workflows, and frankly, traditional methods whether it be manual annotation or nuclear-based AI methods, just have significant limitations in this arena. What was shown here and what I have hoped to have convinced you of is that by incorporating membrane staining like that from molecular instruments into AI-driven segmentation methods that can be developed in the Visiopharm discovery platform, we can achieve more accurate cell boundary detection, improved RNA dot assignments, and increased reproducibility and scalability. And together, all these things represent an important step forward in making spatial biology results more reliable and interpretable. So I'd like to end there, and thank you for your time, and I look forward to being able to answer your questions.
Thank you, Dan and Randy, for those wonderful talks. Before I begin, I just wanted to state that the data that Dan presented is available from a poster that we co-presented at SITC last year. You can find the poster either on the Molecular Instruments website or on the Visiopharm website, or just reach out to us and we'll make sure that you get this information. My talk builds upon what we've just heard about ISH techniques and AI-assisted cell segmentation. We saw how membrane stains improve segmentation accuracy and help assign transcriptomic signals to the correct cellular compartments. We'd like to point out that there's wonderful dot detection that's also going on in this analysis that was just looked over, but there will be a companion piece to this at AACR in a couple of weeks in 2025. So look for that. But now let's take a step further and look at the future of multi-omic imaging as we move from small plex to high plex analysis and how AI will be essential in this transition. The move from small plex to high plex has some considerations when it comes to stain consistency and scan quality. Also, the order of acquisition of the different probes becomes important. And of course, protease-free protocols must be observed if you're going to be analyzing ISH probes and protein at the same time. Consider whether you want to do all of these experiments on the same piece of tissue or in orthogonal experiments through serial sections. Also, as you're considering moving from low to high plex, really consider what signals you're going to be measuring. If you're looking at protein, you're likely studying intensity. Those measurements will be sensitive to the saturation points of the detection systems that you're using. If you're doing ISH, please make sure that the fine focus on those instruments are able to detect dots. And also make sure that you're including the appropriate markers to delineate the different structures that you're interested in, whether those are mapping the tissue, cell segmentation, or even subcellular compartments. Spatial transcriptomics has given us incredible insight into gene expression at the tissue level. However, integrating multiple molecular layers such as protein, metabolites, and even lipidomics within the same tissue sections require new approaches. The challenge is not just data collection, but making sense of this high dimensional information in a way that is biologically and clinically meaningful. One of the key solutions to multi-omic integration is a spatial co-registration technique. By aligning multiple imaging data sets from different modalities into a single tissue image, you can share information amongst those different techniques. Tissue Align by Visiopharm enables this by mapping spatial transcriptomics data with protein imaging and other modalities. This approach provides a richer multimodal view of the tissue microenvironment, helping us link gene expression with cellular phenotypes more accurately. This was done recently in a manuscript delivered by MD Anderson from Sami Ferry Borgogno and Jared Burks, where they were able to get information for 3 different omics: proteomics using Lunaphore Comet, metabolomics using Bruker-Maldi, and transcriptomics using Stomix device from Complete Genomics, where they were able to analyze the molecular, metabolic, and subcellular map of the tumor immune microenvironment. High-plex imaging is not just about increasing the number of markers, it's about handling the exponential rise in data complexity. We now have data sets that contain millions of spatially resolved measurements, which makes manual analysis impossible. This is where AI comes in, not just for segmentation, but for integrating and interpreting multi-omic data at scale. AI assists at multiple levels, from pre-processing and segmentation to advanced pattern recognition. For segmentation, AI ensures accurate cell boundaries, even in densely packed tissues, which is critical when analyzing high-plex data. AI-driven feature extraction allows us to find hidden spatial relationships between transcriptomics and proteomics. And finally, machine learning models can help identify new biomarkers and predict disease outcomes based on the spatial multi-omic data. One powerful application is in the tumor microenvironment analysis. AI can integrate transcriptomics and proteomics to classify different immune cell states within a tumor. This has major implications for immuno-oncology, helping us predict therapy response more accurately. This is a manuscript from Fox Chase Cancer Center, where they were looking at pancreatic ductal adenocarcinoma and the immune response in association with cancer-associated fibroblasts. And what's interesting in this paper was that the fibroblasts that were interacting with immune cells tended to be further away from the tumor than was originally anticipated. As we push multi-omic imaging forward, the ultimate goal is clinical translation. The ability to spatially map gene and protein expression at single cell resolution has potential applications in pathology, drug development, and personalized medicine. And we will need AI to make sense of these massive data sets in a way that clinicians and research can use for decision making. For multi-omic imaging to actually reach the clinic, though, we need to think just beyond the technology. First, integration with other clinical data sources, such as liquid biopsy and genomic sequencing, will be key. Have that orthogonal information. Second, standardization is critical, both in terms of image processing pipelines and regulatory approvals. And third, automation will be necessary to scale these methods for widespread use in hospitals and research labs. At Visiopharm, we think about this with the platforms we develop, where Phenoplex has been designed as a platform for spatial biology, where you can look at as many signals as necessary across those multimodal, multi-omic panels and narrow down from the large list to the pertinent targets. Using our discovery platform, AI can be designed and trained to specifically look for those targets and their spatial relationship within different diseased tissues. Those protocols can be built to be more robust and handle more use cases and understand a larger patient environment where those algorithms can then plug into our decision support tool we call Insight, with those protocols validated and standardized in the clinic. They can also be accompanied by companion diagnostic tests, which will make sure that the samples going into the diagnostic tests are of sufficient quality through quality topics. Now, I imagine a future where spatial multiomics is a routine part of precision medicine. AI-driven analysis could help identify patient-specific biomarkers, guiding therapy selection in real time. We are moving toward a world where tissue imaging doesn't just describe biology, it helps drive clinical decisions. Multiomic imaging is moving from small plex to high plex, requiring new integration approaches. AI is essential for making sense of complex spatial data at scale. And the future is clinical, with multiple pathways for translating these technologies into real-world applications. Thank you for your time. Thank you for attending this three-part series. We'd love to hear your thoughts and take any questions that you may have now. Thank you again.
Hello, everybody. Can I just start by saying sorry for at the beginning? You know, a real webinar is not a webinar if you don't have technical issues. So here it is. But thanks, everyone, for the excellent presentation. I think we can go through the Q&A and, yeah, have a nice discussion. So I can see we got a question from Karen Patel. Does Visiopharm dots detection workflow use AI or is traditional segmentation methods?
It's actually a combination of those two. Getting some feedback. Where the dots are detected via traditional methods and cellular objects are detected via AI. So we're combining those two methodologies.
All right, thank you. Next one from Fergus. How do you train the membrane-aware object detection? Do you mark up nuclei and whole cell objects as separate label classes for training?
Yes, that's exactly how we do it. So we can start with our pre-trained algorithm for finding nuclei, supplement those labeling methods with membrane labeling, and then push a train button.
Cool. And next one from Les. Thanks for the talk, everyone. I wanted to know if this method can be used on any form of RNA or DNA. For example, can it be used with tRNAs, siRNAs, incRNAs?
Yeah, I'll take this one. So currently, we don't have one that detects DNA. That's in the pipeline. And for RNA, we are also currently working on the short target detection, especially the microRNA and also siRNA. So again, those are in development.
Cool. I do have a question for you, Randy. What are the main advantages of the HCR platform compared to other ISH techniques?
I see. So our technique is very sensitive because due to the robust amplification of the HCR hairpins. And those hairpins are very small, so it can penetrate the tissues a lot better. So as a result, we don't need protease digestion. And the technology is also faster and cheaper and more cost effective.
All right, thank you. Do you guys want to share something? Any comments about the presentation? In the meantime,
I just wanted to thank the audience and thank you, Letitia, for hosting.
Yes, thank you, the audience as well. Mhm. If we don't have more questions, you can always email us and contact the speakers later if something comes up. And we'll be sharing the recording as well. So as you rewatch the session, yeah, if any question comes up, you can reach out to us. I think, okay, I just saw one more here. Are the hairpin methods restricted to certain fluorophores, or can any be used?
No, as long as it complies with the conjugation method, our conjugation method, then it can be used. We have conjugated fluorophores or other haptens onto our hairpin. So if you want to make sure your fluorophores can be conjugated, then just contact us at Molecular Instruments.
All right, thank you. And Fergus is asking, would you recommend using UNet or DeepLab for forming whole cell objects?
So in the data that you saw today, I used the UNet network. So just for those of you that we offer a couple different convolutional neural networks in our platform, UNet being one, DeepLab being another. For the applications you saw today, UNet was sufficient for training the network and training the AI to recognize the objects that you saw today. So, but yeah.
All right, if we don't have any other questions, I will say thank you everyone for joining. And you can expect to hear from us with the recording very soon. And yeah, have a nice day. And thank you, the speakers, for the nice presentations. Yeah. Bye.